Tonight, we are looking at one of the strangest quiet ideas in the natural world. How trees talk. Not with voices, not with words, and not with thoughts moving through the forest the way human thoughts move through speech. A white oak standing in a field has no mouth. A sugar maple turning red in October has no ears. A Douglas fir in an old forest has no brain hidden under its bark. And still, trees are not as silent as they seem. They notice light. They notice water. They notice damage. They notice chemicals in the air and soil. They respond to insects, fungi, microbes, shade, drought, gravity, and the slow changes of the seasons. A tree may stand in one place for 100 years, 200 years, or in the case of some bristlecone pines, more than 4,000 years. But standing still does not mean doing nothing. The body of a tree is always measuring the world. Its leaves measure sunlight. Its roots explore soil. Its bark reacts to wounds. Its buds track cold and warmth. Its tiny leaf pores, called stomata, open and close to trade gases with the air. And under the ground, its finest roots release sugars, acids, and other small compounds into the soil around them. A tree's kind of talking begins with a simple idea. One living thing can change something around it, and another living thing can respond. That is communication in the biological sense. It does not require a sentence. It does not require intention. It does not even require a nervous system. When a leaf is chewed by a caterpillar, the damaged cells can release small airborne chemicals called volatile organic compounds. Volatile simply means they can evaporate into the air. These are the kinds of molecules that help create the sharp green smell of cut grass, crushed leaves, or fresh plant sap. To us, that smell may seem pleasant and familiar. To a plant, it can be part of an alarm. Some of these chemicals drift away from the damaged leaf. Nearby leaves, and sometimes nearby plants, may respond by preparing their own defenses. They may begin making bitter compounds. They may change the activity of certain genes. They may become quicker to react if insects arrive later. This is not a tree shouting a warning. It is chemistry moving through air. And yet, the result can still be meaningful. A signal leaves one body. Another body changes. The same kind of quiet exchange happens below ground, but more slowly, and in a much more crowded place. Tree roots do not push through empty dirt. They grow through soil filled with water films, mineral grains, dead leaves, bacteria, fungi, tiny animals, and old carbon from earlier life. Around each root is a thin zone called the rhizosphere. That word means the small region of soil shaped by a living root. In that zone, roots release compounds known as exudates. These can include sugars, amino acids, organic acids, and other chemicals that feed or influence nearby microbes. Some fungi form partnerships with tree roots. These partnerships are called mycorrhizae, a word that means fungus root. The tree gives the fungus carbon-rich sugars made in its leaves. The fungus sends out thin threads that can reach into soil spaces too small for roots, helping gather water and nutrients such as phosphorus and nitrogen. Some fungal threads may connect more than one plant. That idea has become famous as the Wood Wide Web. It is a useful image, but it must be handled gently. The forest is connected, but not every connection works like a message line. Not every exchange is kindness. Not every underground thread is a secret conversation. The real story is quieter and more interesting. Trees communicate through scent, shade, water stress, root chemistry, fungal partnerships, insect attacks, and the living soil around them. Their signals can be clear or faint, helpful or costly, accidental or repeated, fast as a chemical drifting from a wounded leaf, or slow as a root changing the soil over a season. So when we say trees talk, we are not saying they speak like people. We are saying that a forest is full of information. It moves through air. It moves through roots. It moves through fungi. It moves through light. And the first step in hearing that quiet language is understanding how a tree notices the world at all. A tree receives the world through many small living surfaces at once. High above the ground, every leaf is a thin solar panel, a gas exchange surface, and a light sensor. Below the ground, every fine root tip moves through dark soil, testing moisture, minerals, oxygen, and the pull of gravity. Between those two worlds, the trunk carries water upward, sugars downward, and slow signals through living tissue. This kind of sensing is spread through the body. A tree has no single control room. The leaves, roots, buds, bark, and growing tips each respond to their own part of the environment. A sugar maple leaf in July can measure how much light reaches it through the canopy. A pine root near a dry patch of sandy soil can grow more slowly there, while another root follows a wetter path. A bud on a beech twig can track shortening days in autumn and chilling hours in winter, so it opens at a safer time in spring. The tree's body is constantly turning small measurements into growth. Light is one of the clearest examples. Leaves do more than collect brightness. They also sense color. Sunlight contains many wavelengths, and plants are especially sensitive to red light and far red light. Red light is strongly absorbed by leaves during photosynthesis, the process that uses sunlight, carbon dioxide, and water to make sugars. Far red light passes through or reflects from leaves more easily. So when a young sapling receives less red light and more far red light, that light balance can reveal that other plants are nearby or overhead. The proteins that help detect this are called phytochromes. A phytochrome is a light-sensitive plant protein that changes form when it absorbs red or far red light. That small change inside a cell can influence growth. A shaded seedling may stretch its stem upward. A branch may grow toward a brighter opening. A leaf may change its angle to catch more light. These are slow movements, often measured over days or weeks, but they are responses to information. On the surface of leaves are tiny pores called stomata. Each stoma is like a little adjustable doorway. Carbon dioxide enters through these pores, and the tree uses that carbon dioxide to build sugars. Water vapor also escapes through them. On a warm afternoon, a large tree can move hundreds of liters of water from soil to air through its leaves. During dry conditions, the stomata begin to close. That helps save water, though it also slows the entry of carbon dioxide. The tree is balancing thirst against hunger with thousands or millions of microscopic openings. Buds carry another kind of information. Many trees in temperate forests, such as red oak, sugar maple, and paper birch, use day length and temperature to time their yearly cycle. In late summer and autumn, buds form with tiny folded leaves or flowers. Already inside, A protective scale covers them through cold weather. After enough winter chilling, and after spring warmth returns, those buds can open. The timing matters. Opening too early can expose young leaves to frost. Opening too late can waste good weeks of sunlight. Along the trunk, just under the bark, lies the cambium. This is a thin, living layer that produces new wood toward the inside and inner bark toward the outside. In places with strong seasons, cambium activity creates annual rings. A wet growing season may leave a wider ring. A drought year may leave a narrow one. The trunk quietly records conditions in wood. Roots add another layer of sensing. A root tip has a protective cap at its end, and just behind it are cells that divide and stretch. Roots detect gravity, which helps them grow downward. They also respond to water gradients, oxygen levels, salt, and minerals such as nitrate and phosphate. Fine roots can branch more heavily in a rich patch of soil. They may slow in compacted soil where oxygen is low. They release small amounts of chemicals around themselves, changing the thin root-shaped zone where microbes and fungi gather. Information becomes action throughout the tree. A pore opens, a root bends, a bud waits, a branch stretches, a trunk thickens. A chemical signal moves from one cell layer to another. Hormones carry instructions through sap. Electrical changes can pass across cell membranes. Water pressure shifts through living tissues. The whole body is slower than an animal body, yet highly responsive. That becomes clearest when the signal is sudden. On a summer leaf, a caterpillar lowers its head and bites through the green surface. The bite opens a tiny wound. A caterpillar mouthpart cuts through the waxy outer skin of the leaf and into the green tissue beneath. Inside that tissue, thousands of plant cells are packed with water, sugars, proteins, minerals, and small chemical tools. Their cell walls give the leaf its shape. Their inner membranes hold important materials in place. When the caterpillar chews, those walls break. Cell fluids leak into spaces where they usually do not belong. Fragments of cell wall appear around the wound. Oxygen touches fresh surfaces. Compounds that were separated inside healthy tissue suddenly mix. The leaf begins to treat the bite as information. Some trees and other plants can respond differently depending on what kind of trouble has arrived. A clean tear from wind damage leaves one pattern. A chewing insect leaves another. A fungus entering through damp tissue creates a different set of signals. A dry spell around the roots creates a separate warning through water stress. The difference matters because each problem is expensive to answer. Making defense chemicals takes energy and carbon that could have gone into growth, wood, roots, flowers, or seeds. A tree benefits when it prepares the right defense at the right time. Chewing insects give the leaf special clues. As a caterpillar feeds, tiny amounts of saliva and digestive fluids touch the wounded plant tissue. Some plants can detect compounds in those insect fluids. Those traces help the leaf recognize feeding damage more clearly than a simple scratch. In species studied closely, including poplar relatives and many crop plants, this kind of detection can change which defense pathway becomes stronger. One important pathway uses jasmonic acid. Jasmonic acid is a plant hormone, meaning a chemical messenger made in one part of the plant that changes activity in another part. It is strongly linked with defenses against chewing insects such as caterpillars, beetle larvae, and sawfly larvae. When jasmonic acid rises, cells may begin making chemicals that make leaves harder to digest or less pleasant to eat. Some defenses are tannins. Tannins are bitter compounds that can bind to proteins and interfere with digestion. Oaks, willows, and many other woody plants contain them. Other defenses include phenolic compounds, which are a large group of plant chemicals often involved in protection. Conifers such as pines and spruces may produce sticky resins that can slow insects, seal wounds, and carry strong-smelling defensive chemicals. A separate pathway often involves salicylic acid. This chemical is more closely tied to responses against many pathogens, such as bacteria, fungi, and viruses. The name is related to willow, from the genus Salix, because willow bark contains related compounds that humans have used in medicine for a long time. Inside the leaf, the first alarm can spread in several ways. Calcium ions are one of them. An ion is an atom or molecule with an electrical charge. Calcium ions can move into plant cells after damage, changing the activity of proteins and genes. In many plants, waves of calcium activity can pass from wounded tissue into nearby tissue within minutes. Electrical changes also move through plant cells. Each living cell has a tiny difference in electrical charge across its outer membrane. Damage can disturb that balance. The disturbance can spread along connected tissues, helping carry wound information beyond the bite mark. Plants also use water pressure as part of their internal signal system. A torn leaf changes pressure in nearby veins. Those veins are small transport lines carrying water, dissolved minerals, and sugars. A sudden wound can alter the flow of water through them, adding another layer to the warning. These signals do not all travel at the same speed. Electrical and calcium signals can move quickly by plant standards. Hormones and defense chemicals often build more gradually. A nearby leaf on the same branch may begin changing its chemistry before an insect reaches it. A leaf farther away may receive a weaker or later signal through the tree's transport tissues. The result is a body preparing ahead of the damage. Cells that have never been bitten may start producing bitter compounds. Proteins that interfere with insect digestion may increase. Some tissues may become less nutritious. Others may thicken or seal around wounds. The tree is using part of its stored sunlight to defend the rest of itself. This is the first kind of tree message. It begins inside one leaf, then moves through living tissue. The message has no sound, yet it changes what the tree becomes ready to do. Around the wound, some of the new chemistry stays in the cells. Some travels through veins and sap. A few lighter molecules begin to lift away from the torn green surface and enter the air. The lightest wound chemicals rise first. They pass through tiny openings in the leaf surface and spread into the still air around the branch. A person walking past a freshly cut lawn knows one version of this signal as a clean green smell. That scent comes from injured grass cells. The same kind of chemistry can rise from crushed clover, broken maple leaves, trimmed tomato vines, or a torn poplar leaf after an insect bite. The sharp green odor is partly made by green leaf volatiles. Volatile means the molecules evaporate easily. A puddle slowly disappears into air because water is volatile enough to become vapor. Plant volatiles do something similar, though many are oils or small carbon-based chemicals rather than water. Green leaf volatiles often appear within seconds or minutes after damage. They include compounds with names such as hexanol, hexanol, and hexanyl acetate. those names are less important than what they do. They are small enough to leave the damaged tissue, light enough to travel, and reactive enough to become part of the plant's first outside signal. The smell of fresh-cut grass is pleasant to many people because it is familiar. Inside the plant world, that smell often begins with broken cells. A leaf does not release a single chemical word. The damaged surface releases a changing blend. At first, fast wound chemicals may rise quickly. Later, other compounds may increase as the leaf's defense system becomes more organized. Terpenes are one major group. Many trees make terpenes, especially conifers such as pine, spruce, fir, and cedar. Terpenes help create the resinous smell of pine needles and sticky sap. They can also play roles in defense, attraction, and communication. Methyl jasmonate is another airborne compound linked with stress signaling. It is related to jasmonic acid, the hormone involved in many chewing insect defenses. Methyl salicylate can also appear in plant stress responses. Its scent is familiar because it is related to wintergreen. In plants, it can be connected with defense pathways that often respond to pathogens and certain kinds of injury. When insects feed, researchers often call the released scent mixture herbivore-induced plant volatiles. Herbivore means plant eater. Induced means caused or triggered. So the phrase simply means airborne plant chemicals released after an animal feeds on the plant. A caterpillar chewing a willow leaf may produce one chemical pattern. A beetle larva feeding under bark may lead to another. A fungus entering wet tissue may push the chemistry in a different direction. Drought can change leaf chemistry as well, because closing stomata, slowed photosynthesis, and water stress all change what the tree can make and release. The blend can shift over minutes and hours. Early compounds mark fresh injury. Later compounds may reflect hormone changes, defense gene activity, and new chemicals being built inside cells. A tree's scent after damage is closer to a changing recipe than a fixed label. The surrounding weather also shapes the message. Warm sunlight can increase evaporation from leaf surfaces. Cool evening air may slow it. High humidity can change how molecules move and settle. Wind can stretch an odor plume into a thin ribbon, or break it into patches. Rain can wash some compounds from leaves and change the surface where they form. A smooth, waxy leaf may hold and release molecules differently than a hairy leaf. Even leaf age matters, because young leaves and older leaves often contain different amounts of water, wax, nitrogen, and defense chemicals. Distance changes everything. Close to the wound, the scent is strongest. A few centimeters away, it begins to thin. Across several meters, wind and mixing can make the signal uneven. In a dense forest, thousands of leaves, needles, flowers, fungi, wet logs, and soil microbes add their own odors to the air. A single tree's signal enters that shared mixture. The forest air becomes a moving chemical space. A red oak leaf, a balsam fir needle, a birch twig, and a patch of crushed fern may all contribute different molecules at the same time. For the injured tree, releasing these chemicals may help coordinate its own defenses. Nearby leaves on the same branch may receive traces. Other branches may be exposed as air moves through the crown. The signal has crossed an important boundary now. It began as damage inside living tissue. It became scent. it entered the space between plants. Out beyond the wounded leaf, another surface waits in the path of the drifting molecules. A nearby leaf does not need to touch the wounded branch to be affected by it. Its surface is already open to the air. Tiny pores are taking in carbon dioxide. Waxy layers are holding moisture inside. Cells near the surface are exposed to faint traces of the chemical world around them. When damage-related molecules drift across that surface, some may dissolve into the thin film of moisture on the leaf. Others may enter through stomata or settle on the waxy outer layer. Inside the receiving plant, those traces can begin small changes. The plant may adjust the activity of certain genes. A gene is a stretch of instructions inside a cell. When a gene becomes more active, the cell may begin making more of a certain protein or chemical. This is one way a plant can prepare before damage reaches it. The useful word here is priming. Priming means the plant gets ready to respond faster or more strongly later, while holding back from a full defense response right away. That matters because defense is costly. A leaf filled with bitter compounds may be safer from insects, yet those compounds require carbon, nitrogen, and energy. A tree that spends too much on defense during a quiet season may grow less wood, make fewer roots, or produce fewer seeds. Priming gives the plant a middle path. Its cells become alert. The full chemical cost can wait until the threat becomes more direct. Researchers have observed this kind of effect in several plant systems. Willows have been studied because their leaves are eaten by many insects, including beetles and caterpillars. Poplars, which are close relatives of willows, have also been important in tree defense research. Black alder, a moisture-loving tree often found near streams and wetlands in Europe, has appeared in studies of airborne cues and insect feeding. Sagebrush has been especially useful in field studies of plant scent, even though it is a shrub rather than a tree. Tomatoes and lima beans have helped scientists study these signals in controlled greenhouse settings, where airflow, insect damage, and plant spacing can be measured more carefully. Across these systems, the pattern is careful and specific. In some cases, plants exposed to herbivore-induced plant volatiles later show stronger defense responses. Herbivore-induced plant volatiles are airborne chemicals released after a plant eating animal feeds. A receiving plant may increase jasmonate-linked activity, which is connected to defenses against chewing insects. Some plants produce more phenolic compounds. Others may build more tannins, the bitter chemicals common in oak leaves, tea leaves, grape skins, and many woody plants. Some make protease inhibitors. A protease is an enzyme that helps break proteins apart during digestion. A protease inhibitor interferes with that process. For an insect larva eating leaf tissue, that can make the meal harder to use. The leaf may still look the same from the outside. Its color may stay green. Its shape may remain smooth. Deep inside the cells, the plant has changed the way it is prepared. A nearby caterpillar arriving hours or days later may find a less useful meal than it would have found before the warning scent passed through the air. The receiving plant has no picture of the wounded neighbor. Its cells do not know there is a willow branch, a poplar leaf, or a caterpillar nearby. The response comes from chemistry meeting living tissue. A molecule reaches the surface. A receptor or chemical pathway reacts. Genes change activity. New compounds begin to form. That chain can still carry information, because the first event affects the second. The signal can also move within the same tree. A damaged leaf on one branch may release volatiles that reach leaves higher in the crown. In a still orchard or a dense young forest, leaves from the same plant and leaves from nearby plants may share the same pocket of air for a while. Spacing matters. A seedling 10 centimeters away receives a stronger trace than one several meters away. A calm morning may hold the scent close. A gusty afternoon may break it apart. This is why plant communication through air is measurable, useful, and uneven. It can prepare nearby tissues. It can shift insect feeding. It can change the timing of defense. The air carrying it remains restless, filled with odors from bark, soil, flowers, wet leaves, fungi, and many other living surfaces. A scent moving through forest air rarely travels alone. The wounded leaf releases its own blend, and that blend enters air already carrying traces from bark, needles, flowers, damp moss, fallen leaves, soil fungi, and living roots. A single damaged plant can release dozens of volatile compounds at the same time. Some rise almost at once from broken cell membranes. Others build more slowly as hormones turn on defense pathways. A poplar leaf chewed by an insect may release green leaf volatiles first, then terpenes and other stress-related compounds later. A pine seedling under attack may add resin-scented molecules to the air. A willow branch beside a stream may release one pattern during fresh chewing damage and a different pattern during drought or infection. The useful picture is a chemical cloud. It has edges that shift. It has stronger and weaker patches. It changes as the damaged tissue changes. Each molecule behaves a little differently. Some are light and move easily. Some react quickly with sunlight or other chemicals in the air. Some cling more readily to leaf waxes, bark cracks, or damp surfaces. Ozone, a reactive form of oxygen found in small amounts near the ground, can break down certain plant volatiles. That means air pollution can shorten the life of some scent signals before they travel very far. Distance also changes the message. Close to the damaged leaf, the signal may be concentrated enough for nearby tissues to detect clearly. A meter away, the same blend may be weaker. Farther through a thicket, it may arrive in scattered traces, depending on the movement of air between stems and leaves. Wind is one of the strongest forces shaping the signal. A light breeze can carry an odor plume along a branch or across a gap between two saplings. A stronger gust can stretch that plume into a thin streak. Turbulent air can break it into uneven pockets, so one leaf receives a brief pulse, while another leaf nearby receives almost nothing. Foresters and ecologists sometimes think about this the same way insect researchers think about flower scent. An odor in open air moves more like smoke from a candle than like a straight line. Temperature matters as well. Warm leaves often release volatiles more easily than cool leaves. On a sunny afternoon, the surface of a leaf may become several degrees warmer than the surrounding air. That warmth can increase evaporation from waxy surfaces and open tissues. In cooler evening air, the same chemicals may move more slowly. Humidity changes the journey too. Moist air can affect how molecules settle onto surfaces. Rain can wash compounds from leaves and bark. After a shower, a forest may smell stronger to a person walking through it, partly because water has changed the release and movement of many plant and soil compounds at once. The shape of the receiver matters as much as the shape of the signal. a smooth magnolia leaf, a hairy mullein leaf, a waxy pine needle, and a thin birch leaf all give airborne molecules different surfaces to meet. Leaf waxes can absorb some compounds. Tiny droplets of moisture can hold others for a short time. A nearby plant may take in an airborne chemical, change it inside its tissues, and store it in a less active form until it is needed. Some plants exposed to certain green leaf volatiles can later convert those molecules into defensive products. The signal's effect depends on species, timing, dose, and past experience. A young sapling already stressed by low water may respond differently than a healthy one in rich soil. A leaf that has been bitten before may react more strongly than a leaf meeting the scent for the first time. A chemical trace that prepares one species may mean little to another. The same forest air can hold a useful warning, a confusing background odor, and a clue for some other living thing. A moth looking for the right plant may follow part of the scent. A beetle may avoid a leaf that smells strongly defended. A tiny wasp may use the odor of chewed tissue to find the caterpillar feeding there. The message has moved beyond the tree that released it. It now belongs to the shared air, where every listener receives it through a different body. A scent that prepares one leaf can guide a very different body. For a plant, the odor of chewed tissue may carry warning. For an insect, the same odor may point toward food, danger, or a place to lay eggs. A caterpillar searching for a young leaf does not need to understand the tree. Its antennae carry tiny scent receptors that respond when certain molecules touch them. Many insects use these receptors the way a person might use smell in a kitchen. A faint trace can lead them closer to a source. A stronger trace can tell them they are near the right surface. Plant odors help some insects find the species they can eat. A moth may follow leaf chemicals toward a host plant where its caterpillars can feed. A beetle may respond to the smell of stressed bark. An aphid may settle more readily on a plant that gives off the right blend of green tissue and sap. Yet a strong, damaged smell can also push some insects away. A leaf that already smells heavily attacked may suggest crowding, low food quality, or active defenses. A plant that has begun producing bitter tannins or other protective compounds may become a poorer meal. For an insect, the smell of damage can mean opportunity in one setting and risk in another. That mixed meaning becomes especially important because insects have enemies too. Some of the smallest listeners in the forest are parasitoid wasps. A parasitoid is an insect whose young develop on or inside another insect host. Many parasitoid wasps are only a few millimeters long. Some are smaller than a grain of rice. They can use herbivore-induced plant volatiles to locate caterpillars, aphids, or beetle larvae hidden on leaves. The plant is damaged by the feeder. The damaged plant releases a chemical blend. The wasp follows part of that blend toward the feeding insect. This has been studied in crop plants such as corn, cotton, lima beans, and tomatoes, where caterpillars can be placed on leaves and odor responses can be measured in controlled air streams. The same principle matters in woody plants and forests, even though forests are harder to test. A willow leaf chewed by beetle larvae, a poplar branch browsed by caterpillars, or an oak leaf attacked by insects can release odors that become information for other organisms. Predatory mites can respond to damaged plant odors in some plant systems. Lady beetles and lacewings may use plant and prey odors while searching for aphids. Ground beetles, spiders, birds, and other predators use many cues at once, including movement, shape, sound, and smell. The tree's chemical signal becomes one strand in a wider food web. Conifer forests show another side of this. Pines, spruces, and firs make resin filled with terpenes. These chemicals help create the familiar pine smell. They also matter during insect attacks. Bark beetles, including species that attack pines and spruces, can use tree stress chemistry as part of their search for weakened hosts. Some bark beetles also produce their own pheromones. A pheromone is a chemical signal used between members of the same species. When a beetle finds a suitable tree, its chemical signals may help attract more beetles. The tree responds with resin flow and defensive chemistry if it has enough water and stored energy. In Europe, the large pine weevil is a major pest of young conifer seedlings. It often becomes a problem after clear cutting, when fresh stumps release odors that attract adult weevils. Newly planted pine or spruce seedlings nearby can be damaged as the insects feed on their bark. Here, tree smell, forest management, insect behavior, and young seedling survival become linked in one practical chain. A chemical released from wood can shape where insects gather. The gathered insects can affect which seedlings survive. The surviving seedlings influence the next forest. This is why tree communication reaches beyond tree-to-tree warning. A volatile compound can prepare a neighbor, attract a predator, repel a herbivore, or guide a pest. The effect depends on the listener's body. A leaf receives chemistry through plant tissue. A wasp receives it through antennae. A beetle receives it through sensory organs, tuned by millions of years of feeding and breeding near particular plants. Above the soil, the forest is filled with receivers. Signals drift through leaves and needles, across bark, around flowers, and into the small paths followed by insects. Below that living air, under fallen needles and dark leaf litter, roots enter a slower kind of message field. Under the leaf litter, the tree enters a world that is dark, wet, crowded, and uneven. A trunk may look solid from the outside, yet inside it, water is rising through xylem and sugars are moving through phloem. Xylem is the water-carrying tissue that pulls moisture upward from the roots. Phloem is the sugar-carrying tissue that moves food made in the leaves toward roots, buds, bark, and growing wood. Those two transport systems connect the green crown to the hidden root system below. A mature oak, maple, or beech can have large woody roots that help hold it in place. The most active sensing and absorbing happens through much smaller roots. Fine roots are thin, living roots that often measure less than 2 millimeters across. Many fine roots live for only weeks, months, or a few years, depending on the species and the soil. They grow, branch, die back, and grow again as the tree follows water and nutrients through the ground. At the tips of young roots, soft tissues push between mineral grains, bits of decaying leaves, fungal threads, and tiny pockets of air. A root tip can detect gravity, which helps guide growth downward. Moisture changes its direction. Oxygen levels affect whether the root can breathe. Salt can slow growth. Nitrogen and phosphorus can encourage branching when they appear in useful patches. Nitrogen is needed to build proteins and chlorophyll, the green pigment that helps capture light. Phosphorus is part of the molecules cells use to store and move energy. Roots meet these materials in a very uneven way. Soil is never one smooth substance. A handful of forest soil may contain sand grains, clay particles, tiny stones, decayed leaf fragments, water films, air spaces, and pieces of old roots. One side of a root can touch a damp pore filled with dissolved minerals. The other side can face a dry pocket with little oxygen. A few millimeters away, the acidity of the soil may change. This acidity is measured as pH, which tells how sour or alkaline a substance is. Many forest trees grow best in soils with a pH between about 4 and 7, though different species tolerate different ranges. Root hairs help the tree work inside this patchy world. A root hair is a tiny tube-like extension of a root cell. It can be thinner than a human hair and much shorter. Yet millions of them can greatly increase the surface area touching the soil. More surface means more contact with water and dissolved minerals. Root hairs also create closer contact with microbes and fungal threads. The root surface is alive in another way as well. It releases chemicals. These released compounds are called root exudates. An exudate is something that seeps out. Tree roots can release sugars, amino acids, organic acids, enzymes, phenolic compounds, and small signaling molecules. Sugars provide food for microbes. Amino acids contain nitrogen. Organic acids can help change mineral availability near the root. Enzymes are proteins that speed up chemical reactions. Phenolic compounds can affect microbes, fungi, and nearby roots. The thin soil zone shaped by these exudates is called the rhizosphere. Rhizo means root, and sphere means surrounding area. The rhizosphere may extend only a few millimeters from the root surface. Yet it is one of the busiest places in the forest. In that narrow zone, the tree changes the chemistry of the soil. Microbes respond to the food and signals released there. Bacteria gather near fresh carbon. Fungi grow toward root surfaces. Tiny soil animals feed on microbes and dead matter. Minerals are loosened, transformed, absorbed, or locked away again. A root growing through soil leaves a living chemical trail. The trail changes with season, tree age, drought, insect attack, leaf growth, and stored sugar supply. In spring, when buds open and new leaves expand, roots may receive less sugar for a time because the crown is spending energy above ground. Later in summer, when leaves are making more sugar, roots can feed the soil around them more strongly. The hidden half of the tree is therefore active and selective. It senses small differences. It releases food and signals. It changes the ground it passes through. Around those roots, one group of organisms becomes especially important because their thin bodies can enter soil spaces even finer than the finest root hairs. Those thin bodies belong to fungi. In forest soil, fungi often appear as pale threads running through leaf mold, wrapping around roots, or spreading through a fallen log. The mushroom above ground is only the fruiting part, like an apple on a tree. Most of the fungal body is hidden. It grows as hyphae, which are microscopic threads that push between soil grains, around roots, and through decaying wood. A mass of many hyphae is called mycelium. In a forest, mycelium can spread through places that roots cannot easily enter. Some soil pores are far smaller than a fine root. A fungal thread can pass into those narrow spaces and reach bits of water, phosphorus, nitrogen, and other nutrients held between mineral particles. This is where one of the most important partnerships in plant life begins. The partnership is called mycorrhiza. The word means fungus root. A mycorrhizal fungus connects with a plant root, and the two living partners trade. The tree gives the fungus carbon-rich sugars made during photosynthesis. Those sugars begin in green leaves, where sunlight helps turn carbon dioxide and water into food. The fungus receives some of that carbon and uses it to grow. In return, the fungus helps the tree reach materials in the soil. Phosphorus is especially important. Cells use phosphorus in the molecules that carry energy. It is also part of DNA, the instruction material inside living cells. In many soils, phosphorus is present but difficult for roots to reach because it sticks to mineral surfaces or remains locked in organic matter. Fungal hyphae increase the tree's reach. They can explore a larger soil volume than roots alone. They can release enzymes that help break down complex materials. They can help gather nitrogen, which trees need for proteins, leaves, and chlorophyll. They can also help with water access, especially in tiny films held between soil particles. The tree's root system becomes larger in effect because the fungus is working beyond the root surface. There are different kinds of mycorrhizal partnerships. In many temperate forests, ectomycorrhizal fungi are common. Ecto means outside. These fungi often form a sheath around the outside of a root tip. They may also grow between the outer root cells, creating a close exchange surface. Pines, spruces, firs, oaks, beeches, birches, willows, poplars, and many other forest trees commonly form ectomycorrhizal partnerships. Some of the mushrooms seen in forests come from these fungi. Chanterelles, boletes, amanitas, russulas, and many truffles are connected with tree roots during part of their lives. Another major type is called arbuscular mycorrhiza. Arbuscular fungi enter root cells and form tiny branched structures called arbuscules. An arbuscule looks like a miniature tree inside a plant cell. Its many branches create a wide surface for exchange. The fungus gives minerals to the plant. The plant gives carbon compounds to the fungus. Arbuscular mycorrhizal fungi are associated with a very large share of land plants, including many grasses, crops, wildflowers, and tropical trees. Large mapping projects have begun showing how widespread these underground fungi are because they influence plant nutrition and the movement of carbon into soil. That carbon matters because plants pull carbon dioxide from the air, turn some of it into sugars, and send part of it below ground. Some of that below-ground carbon feeds roots. Some feeds fungi and microbes. Some may later become part of soil organic matter. The bargain is steady, yet carefully controlled. A tree does not give sugar away without limits. A fungus does not gather nutrients without using some for itself. Chemical signals help the partners recognize each other. The root changes as the fungus approaches. The fungus changes as it touches the root. Exchange surfaces form only when both sides allow the relationship to continue. The soil around the root becomes a trading place. Carbon moves from leaf to root to fungus. Minerals move from soil to fungus to root. Water, enzymes, hormones, and small signals shift through the same close contact. A tree's hidden life is therefore partly fungal. Its reach, its nutrition, and some of its responses depend on organisms that are neither leaves nor roots. And once a fungal body is attached to one root tip, its threads may continue outward through the soil, searching for another living root. When a hypha reaches a second root, the partnership can widen. One fungal body may touch the root tips of more than one plant. In that case, the fungus is no longer connected to a single tree. It becomes a possible bridge between living roots. Scientists call this a common mycorrhizal network. Common means shared. Mycorrhizal means fungus root. Network means the fungal threads link more than one plant underground. The structure itself can be real. A fungal mycelium can branch through soil, wrap around root tips, enter root tissues in some partnerships, and continue growing outward. In a young forest plot, one fungus may connect to seedlings growing only centimeters apart. In an older forest, roots from neighboring trees may overlap in the same dark soil layer beneath moss, leaf litter, and decaying wood. The popular name for this hidden connection is the wood wide web. It is a memorable phrase because it turns soil into something familiar. The internet connects computers with wires and signals. A fungal network connects living roots with microscopic threads. The image is useful as a doorway into the science. It becomes less useful when it makes the forest sound too much like a planned message system. A physical connection gives material a possible path. Careful evidence still has to show what moves, how much moves, which direction it moves, and whether the receiving plant actually benefits. That last part is difficult. Forest soil is one of the hardest places to run a clean experiment. A teaspoon of healthy forest soil can hold millions or billions of microbes, depending on the soil and the method used to count them. It can contain root hairs, fungal hyphae, dead cells, water films, mineral grains, tiny animals, and dissolved carbon. When a molecule moves underground, it may travel through fungal tissue. It may move through soil water. It may leak from a root and be taken up by microbes. It may pass through direct root contact. It may remain inside the fungus rather than entering another tree at all. Researchers use special barriers to separate these paths. A mesh barrier can allow fungal hyphae through while blocking roots. A finer barrier can block both roots and larger fungal strands while still allowing water and dissolved chemicals to move. Root exclusion methods help separate the effect of living roots from the effect of fungi. Isotope tracers help follow atoms through the system. An isotope is a version of an element with a different weight. Carbon-13, for example, is a heavier form of carbon. A tree can take in labeled carbon dioxide through its leaves. If that labeled carbon later appears in another plant, in fungal tissue, or in soil microbes, the label gives scientists a clue about where the carbon traveled. The clue still has to be interpreted carefully. Finding labeled carbon near another plant does not automatically prove that one tree fed another tree in a useful way. The carbon may have stopped in fungal tissue. It may have been released into the soil and used by bacteria. It may have reached a seedling in an amount too small to change its growth. This is why several popular claims about common mycorrhizal networks are treated with caution. Scientists have warned that mature trees, preferentially feeding their own seedlings through fungal networks, remains hard to prove. They have also questioned how often these networks improve seedling survival in natural forests. Forest roots and fungal partnerships are real, yet the strongest storybook version often moves faster than the evidence. The careful version is still remarkable. Beneath a stand of spruce, birch, oak, or pine, roots can share fungal partners. Those partners can trade carbon for nutrients. Threads can pass through soil spaces finer than roots can enter. Molecules can move through hidden pathways that a person walking above could never see. The forest floor holds connection, exchange, competition, and uncertainty in the same small layer of earth. A network may carry minerals. It may carry chemical signals in some systems. It may shape which seedlings survive near larger trees. The hidden path is real enough to study and complicated enough to resist simple stories. Among all the possible materials moving through this underground world, one has become the most famous because every tree builds its body from it: carbon. High in the crown, carbon enters the tree through a leaf. The source is carbon dioxide, a gas that makes up a small part of the air. During photosynthesis, the leaf uses sunlight to combine carbon dioxide with water. The result is sugar. Oxygen leaves the leaf as a byproduct, while the sugar becomes fuel, building material, and stored energy. A tree cannot build wood, bark, fine roots, flowers, fruit, seeds, or defensive chemicals without carbon. The trunk of a red oak, the needles of a white pine, and the roots of a young birch are all built largely from carbon that first entered through leaves. After sugar is made, the tree begins moving it. The transport tissue called phloem carries sugar-rich fluid away from the leaves. Phloem can send sugars toward growing shoots in spring. It can send them toward roots during active root growth. It can move them into storage tissues, where the tree keeps reserves for winter, injury, or the next flush of leaves. Some of that carbon reaches mycorrhizal fungi. The tree gives the fungus carbon compounds made from photosynthesis. The fungus uses that carbon to grow hyphae, build cell walls, and search the soil for minerals and water. In many mycorrhizal partnerships, this exchange is central. Carbon travels from air to leaf, from leaf to phloem, from phloem to root, and from root to fungus. That path is already a major movement of information and matter through the forest. The more difficult question begins when a fungal body touches more than one plant. Can carbon from one tree reach another tree through that shared fungal system? Scientists have tried to follow this movement with labeled carbon. A label is a marker that makes an atom easier to track. Carbon-13 is a heavier form of carbon that occurs naturally in small amounts. Carbon-14 is a radioactive form of carbon, meaning it gives off energy as it decays. Both can be used carefully in research to trace where carbon goes. In a typical tracing experiment, a plant may be exposed to carbon dioxide containing the label. Its leaves take in that carbon dioxide. Photosynthesis turns the labeled carbon into sugar. Later, researchers test nearby roots, fungi, soil, or neighboring plants to see where the label appears. This kind of experiment has helped show that carbon can move through plant, fungus, and soil systems. Some studies have reported carbon transfer between plants or seedlings. In young plants growing close together, labeled carbon has sometimes appeared in a neighbor after one plant was marked. Forest studies involving birch, Douglas fir, pine, and other species helped make this idea famous in the 1990s. The results were exciting because they suggested that underground fungal links might carry carbon between trees. The careful interpretation is more delicate. A label appearing near a second plant does not tell the whole story by itself. The amount may be very small. The carbon may be inside fungal tissue rather than inside the receiving plant. Soil microbes may have taken it up. Some could have moved through soil water. Direct root contact or root leakage may explain part of the signal. Even when the receiving plant contains labeled carbon, the amount may be too small to improve growth, survival, or health. A tiny transfer can be measured with sensitive instruments and still have little effect on the tree's life. Scale matters. A mature tree can move large amounts of carbon inside its own body each growing season. Leaves send sugars to new wood, root tips, storage cells, and fungal partners. Compared with that internal flow, a detected transfer to another plant may be a thin trace. The trace can still be scientifically interesting. It shows that hidden movement is possible. It also reminds us that possible movement and meaningful support are two different levels of evidence. Carbon transfer becomes especially hard to interpret in real forests. Seedlings under a large canopy may be limited mainly by light. A shaded seedling might receive a small amount of carbon below ground and still fail if its leaves cannot gather enough sunlight. A young tree in dry soil may need water more than sugar. A fungus connected to several plants may direct resources according to its own needs and rewards. The fungus is a living partner, not a passive pipe. Carbon moving through a fungal network, therefore, belongs to a system of trade, cost, and control. The image of one generous tree feeding another can be comforting. The measured forest asks for a more careful kind of wonder. Carbon may move. The path may involve fungi. The effect may matter in some places, at some times, under some conditions. Each part needs evidence. That care does not make the forest less connected. It makes the connection more real. A hidden transfer becomes most meaningful when we understand who pays, who gains, and what changes afterward. The word help sounds simple, while the living forest is much more exact. Inside a forest, human words can make living relationships sound simpler than they are. Help is one of those words. So are share, mother, friend, sacrifice, and care. They can be useful in a loose way because they give the listener a familiar handle. They can also carry too much human feeling into a system built from roots, fungi, chemistry, sunlight, and survival. A large Douglas fir may influence a young seedling nearby. A beech tree may change the light and soil under its crown. An oak may feed a fungus that also touches another plant. Those effects matter. The reason behind them has to be described carefully. Ecologists use plainer words for these relationships. Mutualism means both organisms benefit from the exchange. The mycorrhizal partnership is often mutualistic. A tree gives carbon-rich sugars to a fungus. The fungus helps the tree reach phosphorus, nitrogen, water, or small soil spaces the roots could not explore as well alone. Both partners gain something useful. Competition happens when two organisms need the same limited resource. Two saplings under a closed canopy may compete for light. Their roots may also compete for water or dissolved minerals. In a dry summer, a shallow-rooted seedling and a nearby mature tree may draw from the same shrinking layer of moist soil. Competition can happen even between related trees, growing from seeds that fell from the same parent. Facilitation means one organism improves conditions for another through its presence or activity. The improvement may happen without any intention. A large tree can shade a seedling during a hot afternoon, lowering leaf temperature and slowing water loss. Its trunk and branches can reduce wind near the forest floor, Its leaf litter can add organic matter as it decays. A fallen log can hold moisture like a sponge and become a nurse log where spruce, hemlock, or cedar seedlings root into softened wood. The older plant has shaped the environment in a way that makes survival easier for another living thing. Parasitism describes a relationship where one organism benefits while the other is harmed. Some fungi attack living trees and take food from them. Armillaria, sometimes called honey fungus, can spread through roots and wood, weakening or killing trees in forests, orchards, and parks. Dwarf mistletoes grow on conifers such as pines, spruces, and firs, drawing water and nutrients from their hosts. These organisms are part of forest connection too. Connection can nourish, drain, protect, weaken, or redirect life. Side effects also matter. A maple leaf falls in autumn because the tree is ending the growing season and recovering useful materials from the leaf. After it lands, that leaf becomes food for fungi, bacteria, springtails, mites, millipedes, and earthworms in places where earthworms are present. The maple did not drop the leaf to feed the soil animals. The soil animals still use it. Over time, their feeding helps return nutrients to the soil. A forest can be full of benefits that arose from ordinary life processes. The fungus in a mycorrhizal partnership also has its own needs. A fungal network may connect more than one plant, yet the fungus is alive for itself. It must grow, repair damage, gather nutrients, and receive carbon. If one plant supplies more sugar, the fungus may provide more nutrients to that partner. If another plant supplies little carbon, the exchange may weaken. This creates a marketplace under the soil, though even that image is only a guide. Carbon, phosphorus, nitrogen, water, and minerals move according to living costs and rewards. Evolution works through survival and reproduction across generations. A trait that helps a tree leave more successful offspring can spread. A trait that helps a fungus grow and reproduce can spread. Generosity belongs to human language. Forest biology is measured by growth, survival, seed production, spore production, and the ability to endure stress. This makes the forest more believable. A tree can affect its neighbors deeply. A fungus can link roots. A seedling can benefit from shade, moisture, litter, or fungal contact. Those facts keep their strength when they are described as effects instead of intentions. The living forest does not need to behave like a human family to be connected. Its connections are older, slower, and often more complicated. Even without human motives, nearby trees and roots still carry clues about identity, distance, relatedness, and condition. Those clues gather most closely around the root surface. In the rhizosphere, every root changes the soil in a slightly different way. A young birch root, an old oak root, a stressed pine root, and a maple root growing after spring leaf out do not release the exact same mixture. Their exudates can shift with species, age, season, drought, mineral supply, insect damage, and the microbes already living nearby. A root growing in rich, moist soil may release a different chemical pattern than a root pushing through dry sand or compacted clay. During active growth, more sugars may leak or be released into the soil. During stress, the balance of organic acids, phenolic compounds, and signaling molecules can change. These chemical traces can act a little like fingerprints. They are not fixed labels. They shift as the tree's condition changes. but they still carry information about what kind of living root is nearby. A neighboring plant may respond by changing how its own roots grow. Some plants grow more roots when they detect competition. Others reduce growth in a crowded patch and send roots in another direction. In small plant studies, roots sometimes respond differently to their own roots, related roots, and stranger roots. This is often called self and non-self recognition. Self means the plant's own body. Non-self means another plant. A root system that can recognize its own roots may avoid wasting energy by competing with itself. Instead of producing extra roots in a place already occupied by its own body, it can keep growing into fresher soil. Kin recognition is related, but more careful. Kin means relatives. Some experiments have found that certain plants change root growth when grown near siblings from the same parent compared with unrelated plants. One well-known example involves sea rocket, a coastal plant in the genus Caequile, which can grow along sandy beaches. In experiments, some sea rocket plants produce fewer competing roots when surrounded by siblings than when surrounded by strangers. Other studies have tested small plants, such as Arabidopsis, a tiny mustard relative often used in laboratories because it grows quickly and has a short life cycle. These results show that plants can respond to chemical identity in some settings. They do not mean every tree in a mature forest recognizes family members in the same way. Large trees are much harder to test. Their roots can spread through many cubic meters of soil. A mature sugar maple or Douglas fir may have roots tangled with fungi, old roots, new roots, decaying wood, and roots from several other species. Digging them up changes the system being studied. Separating one root's chemicals from another root's chemicals in a natural forest is difficult. So tree-scale claims need softer language. Evidence from smaller plants shows what is possible. Forest trees may use some related forms of chemical recognition, but the exact strength, range, and importance are still harder to measure. The soil itself keeps changing the message. Bacteria can eat sugars from root exudates within minutes or hours. Fungi can absorb amino acids or organic acids. Clay particles can hold certain molecules on their surfaces. Water can carry dissolved compounds away from the root after rain. Dry soil can slow movement by breaking the thin water films between particles. A chemical cue released by one root may be transformed before another root ever receives it. Some compounds influence the neighborhood more strongly. Black walnut trees are famous for producing juglone, a chemical found in their roots, buds, nut hulls, and leaves. Juglone can affect the growth of some nearby plants, including tomatoes, apples, and certain garden species, though sensitivity varies widely. This is an example of allelopathy, where one plant releases chemicals that influence another plant's growth. In forests and fields, allelopathy is only one part of a wider story. Roots also compete for nitrate, phosphate, potassium, calcium, water, and oxygen. They meet fungal partners. They avoid poor soil. They grow toward useful patches. The underground neighborhood is therefore shaped by identity and need at the same time. A tree's roots are reading local conditions through chemistry, contact, moisture, and minerals. Above ground, the same tree receives a different kind of neighbor signal through its leaves. Before branches touch, before roots cross, a nearby plant can change the color of the light. A sapling can feel that change long before its leaves are fully shaded. Picture a young sugar maple growing under taller trees in a northern hardwood forest. Its roots are in leaf litter and dark soil. Its thin stem reaches upward through ferns, fallen twigs, and last year's brown maple leaves. Above it, the canopy filters sunlight before the light reaches the sapling's small leaves. To human eyes, shade may look like less light. To a plant, shade also has color. Sunlight contains many kinds of light, including red light, blue light, and far red light. Red and blue light are especially useful for photosynthesis. Chlorophyll, the green pigment in leaves, absorbs much of that light, so the plant can use its energy. Far red light behaves differently. Leaves absorb less of it. They let more of it pass through, and they reflect more of it back into the space around them. That means a plant standing near other vegetation receives a change signal. The direct sunlight has been filtered. Some of the red light has already been used by nearby leaves. More far red light remains. The balance between red and far red light becomes a clue. A low red to far red ratio often means leaves are nearby, either above the plant or around it. The plant receives information before its stem touches another stem. It can detect a neighbor through the color of the light. Phytochromes help make this possible. A phytochrome is a light-sensitive protein inside plant cells. It changes shape when it absorbs red or far red light. That shape change affects chemical pathways inside the plant. Over time, those pathways can alter growth. In a seedling, the response may be stem elongation. The stem stretches upward, trying to reach brighter light before other plants close the gap. This response is often called shade avoidance. It is common in many young plants growing in crowded places. A tree seedling in deep forest shade has to use this response carefully. Stretching upward can help it reach light. Extra height also costs energy and may make the stem thinner. A weak stem can bend, dry out, or break. So the plant balances the signal from light with water, sugar reserves, minerals, and the strength of its tissues. Branches can respond too. A young beech or maple may grow longer shoots toward a small opening in the canopy. A side branch receiving better light may keep more leaves. A shaded branch may slow down, shed leaves earlier, or eventually die back. The tree is placing its limited carbon where light is most useful. Leaf angle can also change. Some leaves tilt or arrange themselves to reduce overlap with neighbors. Others flatten into a position that catches light more efficiently. In a dense stand of young trees, these small changes shape who gets sunlight during the growing season. The light signal changes through the day. Morning light enters from one side of the forest. Midday light filters down from higher above. Late afternoon light slants through trunks and branches. Clouds soften the whole pattern. In spring, before the canopy closes, a forest floor may receive bright light for several weeks. Wildflowers, such as trout lily, spring beauty, and bloodroot, use that brief window before maple, oak, and beech leaves expand overhead. By early summer, the same patch of soil may receive only a small fraction of full sunlight. Tree seedlings live inside that changing map. A shade-tolerant species, such as American beech or eastern hemlock, can survive for years under low light. A sun-loving species, such as trembling aspen or paper birch, usually needs a brighter gap to grow well. The difference helps explain why a fallen tree can change the future of a small patch of forest. When a storm breaks a branch or drops a trunk, sunlight reaches the ground in a new pattern. Saplings already waiting below may begin faster growth. Seeds that were quiet in the soil may germinate. The color and amount of light have changed, and the plants respond. A shadow, then, is active information. It tells a leaf that another leaf is nearby. It tells a seedling how crowded the space has become. It tells a branch whether its position is worth maintaining. Across one tree, those choices are small. Across hundreds of trees, repeated year after year, they begin to build the forest ceiling overhead. That ceiling is layered from top to bottom. At the highest level, mature crowns catch the strongest sunlight. Below them, younger trees occupy the subcanopy, where light arrives in broken patches. Shrubs, saplings, ferns, mosses, and spring wildflowers use the understory and forest floor. Each layer receives a different version of the same day. A red oak crown may spend summer in bright sun. A young sugar maple beneath it may spend those same months in green shade. A hemlock seedling near the ground may receive only a small part of the light that reached the upper branches first. This vertical structure forms because trees respond to light for years. A branch that gathers enough sunlight can keep producing more sugar than it costs to maintain. A branch that remains deeply shaded may lose leaves, slow its growth, and eventually die back. The tree keeps investing in the parts of its crown that return enough energy. Over time, the crown becomes a record of where light has been useful. Different species read that light field in different ways. American beech and sugar maple can survive for many years under a closed canopy. Eastern hemlock is even more patient in deep shade, sometimes waiting as a small tree for decades. Balsam fir seedlings can persist beneath older trees in cool northern forests, growing slowly until a brighter opening appears. Their leaves and needles are built to make use of dim light, so they can keep a small living balance in places where faster species would struggle. Sun-loving trees follow a different strategy. Paper birch, trembling aspen, black cherry, and many pines grow quickly when sunlight is abundant. They often appear after fire, windthrow, logging, flood damage, or another disturbance opens the canopy. Their seedlings usually need a brighter start. In an open gap, they can turn strong light into height, leaves, and new wood quickly. In heavy shade, they often lose the race. A canopy gap changes the information of a forest in a single event. During a storm, a mature tree may snap at the trunk, split at a fork, or pull its roots from wet soil. The fallen crown leaves a hole above the forest floor. Sunlight reaches leaves that had grown for years in dim conditions. Soil warms faster in the day. Rain reaches the ground more directly. Seeds already waiting in the soil may begin to germinate. Saplings that were growing slowly may increase their annual height growth. A small gap may favor shade-tolerant trees already present below. A larger gap may give sun-loving species enough light to establish. The shape of the gap matters. So does its direction, because morning light and afternoon light enter from different angles. A narrow opening in a dense stand may send a bright strip across the forest floor for only part of the day. A wide opening can change the whole small climate near the ground. Foresters sometimes call this the light environment because it includes brightness, color, timing, direction, and shade. The canopy also shows visible signs of trees responding to one another. In some forests, neighboring crowns leave small channels of open sky between them. This pattern is called crown shyness. It has been observed in species such as some eucalypts, black mangroves, and certain tropical trees. The exact cause can vary. Wind may make branches rub against each other, breaking tender tips and keeping crowns apart. Light competition may cause branches to grow away from heavy shade. Insects or leaf diseases may spread more easily when crowns touch, so spacing could sometimes reduce damage. Species growth habits also matter. Crown shyness is a real pattern in some forests, but it should be treated as a visible result, with several possible causes. Forest architecture changes slowly. One growing season can add a ring of wood. 10 seasons can change the balance between two saplings. 50 years can turn a bright opening into young canopy. Several centuries can build an old forest with tall trees, standing dead trunks, fallen logs, root pits, mounds, and many levels of shade. The canopy is therefore built from repeated responses. Leaves sense light. Branches grow toward useful openings. Crowns expand, lose limbs, recover from storms, and adjust beside their neighbors. Every tree is shaped by its own body and by the bodies around it. Light gives the crown its direction. Water decides whether that direction can be followed. Inside every crown, the work of light depends on a hidden river. Water enters through roots, crosses living root tissue, and moves into xylem. Xylem is the tree's water-carrying system. In wood, it is built from long, hollow cells that line up into tiny tubes. In broadleaf trees such as oak, maple, and elm, many of those tubes are called vessels. In conifers such as pine, spruce, and fir, water moves mostly through narrower cells called tracheids. The water does not rise because the tree pumps it like a heart. It rises because leaves lose water to the air. This process is called transpiration. When water evaporates from moist surfaces inside a leaf, it pulls on the water behind it. Water molecules cling to each other, so the pull can travel downward through the xylem, from leaf to branch to trunk to root. A tall tree can keep a continuous column of water under tension, even when the leaves are many meters above the soil. A mature red oak on a warm summer day may move hundreds of liters of water from the ground into the air. A giant sequoia can lift water more than 70 meters above its roots. Every leaf taking in carbon dioxide is also risking water loss. The openings that control this trade are the stomata. A stoma is a tiny pore on the leaf surface, guarded by two curved cells that can open or close. When stomata open, carbon dioxide enters. Photosynthesis can run. Water vapor leaves at the same time. During a wet morning, that exchange may be worth the cost. During a dry afternoon, the cost grows. As soil dries, roots receive less water. The pull in the xylem becomes stronger. Leaves begin losing water faster than roots can replace it. The tree's tissues detect this change through water pressure, cell moisture, and chemical signals. One important chemical is abscisic acid. Abscisic acid is a plant hormone involved in drought response. Roots can produce more of it when soil becomes dry. Leaves can also make it as their own water status changes. When abscisic acid reaches the guard cells around stomata, those cells lose water, and the pores begin to close. Closing stomata helps protect the tree from drying out. It also slows carbon dioxide intake. With less carbon dioxide entering the leaf, photosynthesis drops. The tree saves water while making less sugar. That trade can shape everything else: root growth, fungal exchange, defense chemistry, and the ability to repair damage. Hydraulic signals add another layer. Hydraulic means related to water movement and pressure. A sudden drop in water supply can change pressure through the xylem before chemical signals fully build. In some species, water status can trigger stomatal closing quickly. Abscisic acid may then help maintain that closure or strengthen the response. The exact balance differs among species. A drought-tolerant juniper, a shallow-rooted birch, and a moisture-loving willow do not manage water in the same way. The danger inside xylem is cavitation. Cavitation happens when air bubbles form inside the water column. A bubble can break the chain of water molecules and block movement through part of the xylem. In winter, freezing and thawing can also create bubbles. During drought, the stronger pull on water increases the risk. Some trees can repair limited damage when water returns. Severe drought can leave too many blocked pathways. Branches may die back. Leaves may brown at the edges. Fine roots may stop growing or die. A water-stressed tree can become more vulnerable to insects and fungi. Bark beetles often do more damage when trees cannot produce enough resin. Resin is sticky, energy-rich, and water-dependent in its flow. A healthy pine may push resin into beetle tunnels and slow an attack. A drought-stressed pine may have weaker resin pressure and fewer stored resources for defense. Fungal pathogens can also gain ground when tissues are weakened. Drought changes the tree's signals as well. Leaves may release different volatile compounds. Roots may alter their exudates, sending a changed mixture of sugars, acids, and stress-related chemicals into the rhizosphere. Mycorrhizal partnerships may shift as the tree has less carbon to trade. The whole tree becomes a water-shaped message, from closing pores to quieter roots. Around those roots, the small organisms feeding on exudates receive the change first. The rhizosphere is crowded at a scale the eye cannot see. A thin film of water around a root can hold bacteria, fungal spores, fragments of old cells, dissolved minerals, and tiny chemical trails from the tree itself. A single gram of forest soil can contain millions to billions of bacterial cells, depending on the soil, season, and method used to count them. That small amount of soil may also hold fungal hyphae, microscopic animals, dead leaf particles, and bits of root tissue in different stages of decay. The root is growing through a living crowd. Bacteria are among the most active members of that crowd. They are single-celled organisms, usually far smaller than the cells of a root. Many bacteria feed on carbon released from root exudates. Sugars can be used quickly. Amino acids can supply carbon and nitrogen. Organic acids can change mineral availability near the root surface. When a tree changes what its roots release during drought, insect attack, or new leaf growth, the bacterial community can shift with it. Fungi are present there too, beyond the mycorrhizal partners already attached to roots. Some fungi break down dead leaves and wood. Some live quietly near roots. Others become pathogens, meaning they cause disease. A root growing through damp, low-oxygen soil may face different fungal pressures than a root growing in loose, well-aired soil. Archaea, also live in soil. They are single-celled organisms like bacteria in size, but they belong to a separate branch of life. Some soil archaea are involved in nitrogen cycling. Nitrogen cycling means changing nitrogen from one chemical form to another, which affects whether plants and microbes can use it. Protists add movement to this small world. A protist is often a single-celled organism with a more complex cell structure than bacteria. Many protists feed on bacteria. As they graze, they can release nutrients back into the soil in forms roots and microbes may use. Nematodes, which are tiny roundworms, also live among soil particles and water films. Some eat bacteria, some eat fungi. Some attack roots. A few plant-feeding nematodes can pierce root cells and draw out their contents, weakening crops, seedlings, and forest plants. Microarthropods complete the scene at a slightly larger, but still tiny scale. These include springtails and mites, small joint-legged animals that move through leaf litter and soil pores. They shred organic matter, feed on fungi, and help mix small pieces of decaying material into the soil. Their activity changes where microbes grow and how nutrients move. Root exudates help decide which organisms gather closest. A root releasing more simple sugars may support fast-growing bacteria. A root releasing more phenolic compounds may favor different microbes or slow certain organisms down. Tree species matter as well. Oak leaves contain tannins that can influence decomposition. Pine needles add resinous compounds and often create a more acidic litter layer. Sugar maple leaves decay faster than many oak leaves and can support a different soil community. Over years, the chemistry of fallen leaves helps shape the microbes around the roots below. The soil's physical condition keeps changing the conversation. Oxygen is higher in loose soil with open pores. Waterlogged soil can become oxygen poor. Acidity, often measured as pH, affects which microbes thrive. Cold soil slows microbial activity in early spring. Warm, moist soil can speed up decomposition and nutrient release. Dry soil breaks the water films that many microbes need to move, feed, and exchange chemicals. Microbes can help a tree. Some make phosphorus easier to absorb. Others transform nitrogen into useful forms. Certain bacteria and fungi compete with pathogens by taking space or food near the root. A few can prime the plant's immune responses, which means the tree becomes quicker to defend itself after a later threat. The same neighborhood can also harm the tree. Pathogens may use root chemicals as guides toward living tissue. A stressed root releasing a changed exudate blend may attract different organisms than a healthy root. A fungus that was harmless in one condition can become damaging when drought, flooding, or injury weakens the tree. Every root signal is therefore handled by many lives before it reaches another root. A sugar molecule may be eaten by bacteria. An organic acid may loosen a mineral. A fungal thread may absorb an amino acid. A nematode may feed on the bacteria that gathered there. The message from the tree becomes part of a shared underground metabolism. Tree communication in soil is never only tree to tree. It passes through microbes, fungi, tiny animals, minerals, water, oxygen, and decay. This crowded language is very old, shaped by the long history of plants solving the same basic problem: how to survive while rooted in place. Rooted life has been solving that problem for hundreds of millions of years. A plant cannot step away from a hungry insect. A seedling cannot move its stem into a better patch of soil after its roots have taken hold. A tree cannot leave a dry hillside during a drought or walk out from under a crowded canopy. Its body has to sense, adjust, defend, and grow from one place. That simple fact shaped much of plant evolution. The earliest land plants began spreading across wet shorelines and rocky surfaces more than 450 million years ago. Their ancestors lived in water, where drying out was less of a problem. Land offered sunlight and carbon dioxide, but it also brought new dangers. Air could pull water from soft tissues. Ultraviolet light could damage cells. Gravity made upright growth harder. Nutrients were locked in rock, sand, and thin early soils. Microbes and fungi already lived in those places, breaking down minerals and organic matter at small scales. For the first plants on land, partnership was one way to survive. Fungal relationships appear very early in the fossil record of land plants. The Rhynie chert, a famous fossil deposit in Scotland from about 407 million years ago, preserves tiny early plants with fungal structures inside their tissues. These plants were only a few centimeters tall, yet their fossils show a pattern that still matters in forests today. Plants made carbon compounds using sunlight. Fungi helped them reach minerals and water. The ancient bargain between root-like plant tissues and fungi became part of how green life spread across land. As plants grew taller and more complex, their problems changed. Leaves made more sugar, but also attracted more feeders. Stems lifted plants toward light, but exposed them to wind and drying air. Roots reached deeper into soil, but entered a crowded world of microbes, fungi, and competing plants. A plant that could detect trouble early had an advantage. Defenses are useful, but they are costly. A tree that fills every leaf with strong chemicals all season spends carbon and nitrogen that could have built new roots, seeds, or wood. Tannins, resins, bitter compounds, waxes, and protective proteins all require materials and energy. A pine producing resin against bark beetles must use stored carbon. An oak building tannins into its leaves must spend resources gathered through roots and sunlight. A willow making leaf chemicals after caterpillar damage is changing its budget. Priming helps solve this cost problem. When a plant is primed, its cells are prepared for defense without running the full response at maximum strength. The plant may adjust gene activity. It may build small amounts of signal chemicals. It may make certain defense pathways easier to activate later. If the insect attack comes, the response can rise faster. If the attack never comes, the plant has spent less than it would have spent on constant defense. Airborne cues can evolve in that same practical setting. When nearby damage often predicts danger, a plant that reacts to damage-related scent may survive better. A caterpillar chewing one sagebrush branch may mean more caterpillars are nearby. A beetle feeding on a willow may mean other leaves in the same patch are at risk. A chemical released by damaged tissue can become useful information when other plants or other parts of the same plant respond to it. Roots face their own version of the same pressure. Once a root enters soil, it meets neighbors that may need the same water, nitrate, phosphate, and oxygen. Recognizing its own roots can reduce wasted competition. Responding to stranger roots can shift growth toward open soil. Following moisture or minerals can help the plant place fine roots where they matter most. Fungal partners added another layer, because the plant had to regulate a living exchange. Carbon sent below ground needed to return value through nutrients, water access, or protection. These systems were shaped over generations. A tree does not plan them. A fungus does not build them for the beauty of the forest. Useful responses leave more surviving seeds, spores, roots, and shoots over time. The result is a world where scent, shade, touch, water pressure, hormones, root chemistry, and fungal contact all carry information. Long before instruments could identify these signals, people living near forests noticed that trees, soil, fungi, water, and animals behaved as one connected place. Those older observations became sharper when people began to measure forests with instruments. People living close to wooded landscapes have long understood that a forest behaves as a joined place. Many indigenous fire practices, for example, treated trees, grasslands, animals, soil, and seasonal burning as connected parts of land care. In parts of Western North America, low-intensity cultural burning helped maintain open oak woodlands, berry patches, and travel corridors. In parts of Australia, long traditions of controlled burning shaped plant communities and reduced fuel buildup under certain conditions. These practices were based on careful attention over generations. They did not require a laboratory to notice that trees changed after fire, that animals followed new growth, or that fungi and plants appeared after rain. Formal forestry developed with a different focus. In Europe, during the 18th and 19th centuries, forestry schools and state forest services became deeply interested in timber supply. Foresters measured tree height, trunk diameter, stand density, growth rate, and harvest volume. Stand density means how many trees grow in a given area. A stand is simply a patch of forest treated as one unit. Forestry also tracked pests, diseases, spacing, age classes, and the yield of useful wood. Those measurements mattered for building ships, homes, railroads, paper, and fuel. A tree was often studied as a future log, and a forest as a managed crop of trunks. That view gave people useful numbers. It could also miss the quieter exchanges below the leaves and beneath the soil. Modern forest ecology widened the picture. A forest became a system of nutrient cycling, decay, disturbance, water flow, insects, fungi, microbes, seed banks, dead wood, canopy gaps, and living roots. A fallen log became more than waste. It could be a nursery for hemlock or spruce seedlings. A dead standing tree could feed beetles, woodpeckers, fungi, and bacteria. Leaf litter could become a slow pathway by which minerals return to roots. New instruments helped make the hidden parts measurable. Gas chromatography can separate a plant scent into its chemical pieces. Chromatography means separating a mixture as it moves through a material. A crushed leaf may release many compounds at once, but the instrument can spread those compounds into a pattern. Mass spectrometry can help identify them by measuring the mass of charged particles. Together, these tools allow researchers to name chemicals in the scent of damaged willow leaves, pine needles, tomato plants, or flowers visited by insects. Isotope tracers made underground movement easier to follow. An isotope is a version of an element with a different weight. Carbon-13, nitrogen-15, and deuterium which is a heavier form of hydrogen, can be used to trace movement through plants, fungi, water, and soil. A leaf can take in labeled carbon dioxide. A root can receive labeled water. Later, scientists can look for that label in roots, fungal tissue, nearby plants, or soil microbes. The label does not tell the whole story by itself, but it gives the invisible movement a mark. DNA sequencing changed the soil story as well. A small soil sample may contain thousands of kinds of bacteria and fungi. Many cannot be easily grown in a dish. Sequencing allows researchers to identify organisms from pieces of genetic material left in the soil. This revealed that roots are surrounded by communities far more diverse than older tools could show. Sensors made the living tree easier to follow through time. Sap flow sensors can estimate how quickly water moves through a stem. Soil moisture probes record drying and re-wetting after rain. Dendrometers measure tiny changes in trunk size, sometimes showing daily swelling and shrinking as water moves in and out of tissues. Electrical sensors can detect changes after damage, drought, or shifts in light. The quiet forest became a place of graphs, labels, chemical peaks, and tiny measurements. These tools did not remove the wonder. They made the wonder harder to fake. They showed that trees release airborne signals, trade with fungi, shape microbial neighborhoods, and respond to water and light with fine control. They also showed that some beautiful stories run ahead of what has been proven. A measured forest asks for patience, because connection is real while its exact meaning can be difficult to pin down. Patience matters most around the most famous underground story. The Wood Wide Web is a powerful image because it feels easy to picture. Fungal threads run through soil. Roots meet those threads. Different trees may share some fungal partners. A hidden network seems to appear under the forest floor. That basic structure can happen. Common mycorrhizal networks have been found in some plant systems, especially where fungal hyphae connect roots from more than one plant. The harder part is deciding what that connection means. A careful forest story has to separate several layers. First, the pathway itself may exist. Second, material may move through or near that pathway. Third, the movement may change the receiving plant in a useful way. Fourth, the effect may be large enough to matter in a living forest. Those layers can be easy to blend together. A fungal thread between roots sounds like communication. A trace of carbon in a nearby plant sounds like support. A seedling growing near an older tree sounds like care. The science becomes stronger when each claim is tested on its own. Mature forests make that testing difficult. A greenhouse pot can be controlled. A forest floor cannot be controlled as cleanly. Under a Douglas fir, spruce, beech, birch, or oak, roots overlap in tangled layers. Fine roots die and regrow. Fungi pass through soil pores. Rainwater carries dissolved compounds. Bacteria consume sugars. Dead leaves release carbon as they decay. A researcher trying to follow one signal must separate all of those roots. A molecule found near a seedling may have moved through fungal tissue. It may have leaked from a root into soil water. It may have been eaten by microbes, changed into another compound, and released again. A root may have touched another root directly. A mesh barrier used in an experiment may block roots, while allowing fungal threads through. Yet the barrier can still disturb water flow, oxygen, root growth, or microbial movement. Even careful experiments leave room for several explanations. Carbon transfer is one example. Labeled carbon can show that carbon moved from one place to another. The label alone cannot show that a mature tree intentionally fed a seedling. It cannot show that the carbon improves survival. It cannot show that the amount was important compared with the seedling's own photosynthesis. A small transfer may be real and still be too small to change the young tree's future. Defense signals through fungal networks need similar care. In some controlled plant systems, stress signals can move through shared fungal connections and affect nearby plants. That is an important finding. A mature forest adds more layers. Mixed species, changing weather, older roots, many fungi, uneven soils, insects, and microbes that can transform signals along the way. A signal that works clearly in a pot may become weaker, redirected, or hard to detect outside. The language around older trees also needs care. People often speak of mother trees sending food or warnings to their young. The image is memorable, and some older trees certainly shape conditions around seedlings. They change shade, moisture, leaf litter, fungal communities, and soil temperature. Those effects can help nearby young trees in some conditions. Preferential feeding of offspring through fungal networks remains much harder to prove. A seedling below its parent may be there because seeds fell nearby. It may share fungi because roots are close. It may grow better because the forest floor is cooler and moister. Several true effects can exist at once without meaning the same thing. Scientific stories can also become uneven because exciting results travel faster. A clear positive result may be repeated in articles, lectures, books, and documentaries. Mixed results receive less attention. Failed replications may stay quiet. Over time, a cautious finding can become a stronger claim in popular retellings. By the early 2020s, some ecologists were warning that several common claims about forest mycorrhizal networks had grown beyond the evidence. That warning does not erase the hidden forest. It sharpens it. Fungal partnerships still matter. Root exudates still feed microbial neighborhoods. Volatile chemicals still move through leaves and air. Light, water, insects, fungi, and soil still shape how trees respond to one another. The careful version gives each process its proper weight. A connected forest does not need to be simple to be beautiful. It becomes more useful when the connections are measured clearly, because forest care depends on what is actually happening underground and above it. Forest care starts to look different when the hidden systems are included. A woodland is made of trunks, crowns, seedlings, fallen logs, fungi, soil pores, insects, water films, bacteria, and old leaves breaking down on the ground. A planting plan that counts only tree stems can miss much of what keeps those stems alive. The signals between trees and the life around them are part of forest strength. Airborne chemicals can affect insect behavior. Root exudates can shape microbes. Fungal partners can help roots reach phosphorus, nitrogen, and water. Shade patterns decide which seedlings can wait and which seedlings need open sky. Water stress changes all of those relationships at once. This is one reason mixed forests often behave differently from single species stands. A plantation of one tree species, all planted close together and often at the same age, can be efficient for timber. It can also create a simpler target for certain pests or diseases. When many similar trees stand side by side, An insect adapted to that species may move through the stand more easily. A mixed forest has more variety in leaf chemistry, bark thickness, timing of bud break, root depth, crown shape, and fungal partners. That variety can slow some pests, confuse some host-finding insects, or support more predators and parasitoids. The effect depends on the pest and the species mix. Some insects are generalists and feed on many plants. Others specialize on one group, such as spruce, pine, oak, or poplar. Genetic diversity matters inside a species too. Two oaks of the same species may produce different levels of tannins, different leaf timing, or different resistance to drought. A stand grown from a narrow seed source may respond more evenly to stress. A stand with broader genetic variation may contain some trees better suited to heat, insects, frost, or dry years. That variation becomes a quiet form of insurance. Forest managers also think about older trees in a wider way now. A large old tree may provide seed. Its crown may cool the ground. Its leaf litter may shape fungi and microbes. Its trunk may hold cavities for birds, bats, and small mammals. When it dies, standing dead wood can support beetles, woodpeckers, fungi, and nesting animals. After it falls, the log can hold water, protect seedlings, and feed decomposers for decades. Deadwood is part of the living system because decay returns minerals to the soil. Leaf litter works the same way, more gently and more quickly. A maple leaf, oak leaf, pine needle, or beech leaf falls with its own chemistry. Fungi and bacteria break it apart. Tiny soil animals shred it. Rain carries dissolved materials downward. Roots and microbes eventually reuse some of those materials. Removing too much litter, compacting soil with heavy machines, or repeatedly disturbing the same ground can weaken that cycle. Soil compaction is especially important. When soil is pressed down, the air spaces between particles shrink. Roots need oxygen to respire, which means to release energy from sugars. Fungal hyphae also need open spaces and water films to grow through. Compacted soil can slow fine root growth, reduce drainage, hold less oxygen, and make it harder for mycorrhizal fungi to spread. A forest can look green above ground, while its underground exchange system is stressed. Drought and warming add more pressure. Hotter summers can increase water loss from leaves. Dry soil can close stomata and slow photosynthesis. Less sugar made in the crown can mean less carbon available for roots and fungi. Drought-stressed conifers may produce less resin pressure against bark beetles. Some bark beetle outbreaks grow worse when warm temperatures help insects survive and drought weakens tree defenses. Fungal communities can shift as soils warm or dry. Seedlings can fail when heat, low moisture, and poor fungal support arrive together. Restoration, therefore, begins below the surface as much as above it. Planting native tree species can matter. Protecting local seed sources can matter. Leaving leaf litter, old roots, nurse logs, and intact soil structure can matter. In some places, restoration may need to consider mycorrhizal partners already present in the soil. Recent global mapping of arbuscular mycorrhizal fungi has made this hidden scale easier to picture. These fungi are associated with a very large share of land plants, and their threads help move nutrients and carbon through soil. The maps remind us that the underground forest is not a small detail beneath the real forest. It is part of the forest's working body. A tree's messages are useful because they connect survival to place. The air around leaves, the soil around roots, the fungi between particles, and the insects moving through branches all help decide what a forest can endure. After all of this, the word talk returns with more care. It carries scent, shade, water, chemistry, pressure, and time. With that fuller meaning, a quiet forest becomes easier to hear. A person standing beneath an oak, spruce, maple, pine, or beech may notice only wind moving through leaves. The tree is also moving information through forms that are slower and smaller than sound. A chewed leaf can release volatile organic compounds into the air. Those small airborne chemicals may carry traces of injury from one branch to another, or from one plant surface to a nearby leaf. A neighboring plant may become primed. Primed means prepared to respond more quickly if damage arrives later. The change may happen through defense genes, jasmonic acid pathways, bitter tannins, phenolic compounds, or proteins that make an insect meal harder to digest. The signal can also reach other listeners. A parasitoid wasp may follow the scent of damaged leaves toward a caterpillar. A beetle may use tree stress chemistry while searching for a weakened host. A plant-eating insect may avoid a leaf that smells heavily defended, or move toward a scent that matches its food plant. The same chemical trace can pass through several bodies and create several different effects. Below ground, roots create another layer of meaning. Fine roots and root hairs pass through soil only a few millimeters at a time, touching water films, mineral grains, oxygen pockets, and decaying leaf fragments. They release exudates into the rhizosphere, the thin root-shaped zone of soil affected by living roots. Those exudates may include sugars, amino acids, organic acids, enzymes, phenolic compounds, and small signaling molecules. Bacteria may consume them. Fungi may absorb them. Clay particles may hold them. Rainwater may carry them into another pore. The root's message becomes part of soil chemistry before another root ever receives it. Mycorrhizal fungi deepen that exchange. A pine, oak, beech, birch, or spruce can trade carbon-rich sugars with fungi attached to its roots. The fungal hyphae extend into narrow soil spaces, gathering phosphorus, nitrogen, and water that roots may not reach as easily alone. Arbuscular mycorrhizal fungi form tiny branched structures inside root cells. Ectomycorrhizal fungi form sheaths around root tips and grow between outer root cells. Some fungal threads can connect more than one plant, creating common mycorrhizal networks. Those networks are real in some systems, and their exact role in mature forests needs careful evidence. Connection, movement, benefit, and long-term importance are separate things. That careful view gives the forest more depth. Light carries information too. Leaves absorb red and blue light for photosynthesis. They reflect and transmit more far-red light. A young sapling receiving a low red to far-red ratio can detect nearby vegetation before its stem touches another plant. Phytochromes inside its cells sense that light balance. Growth may shift upward, branches may favor brighter openings, and leaves may change their position. Across many years, those small responses help shape the canopy itself. Water adds another form of signal. A tree's xylem carries water upward under tension as leaves lose moisture through transpiration. Stomata open to let carbon dioxide enter and closed to reduce water loss. During drought, abscisic acid and hydraulic signals help close those tiny pores. The tree protects water while slowing sugar production. If the tension grows too strong, cavitation can form air bubbles in the xylem and block water movement. A drought-stressed tree may change its root exudates, reduce carbon sent to fungi, release different leaf volatiles, and become more vulnerable to bark beetles or pathogens. In this way, tree communication is distributed across the whole body. Leaves, roots, bark, buds, fungi, microbes, insects, light, water, and soil all take part. A signal may warn. A signal may prime. A signal may attract, repel, feed, compete, confuse, or reshape conditions for another organism nearby. The forest's language is conditional. Its meaning depends on species, distance, weather, season, soil moisture, past injury, and the bodies receiving the signal. Trees operate without human speech, human thought, or human planning. Their communication is chemical, physical, and environmental. It unfolds through scent rising from wounds, shade changing light color, water pressure shifting in wood, fungal threads trading with roots, and microbes transforming signals in dark soil. The opening image of a silent forest can stay quiet. The quiet now holds more. A leaf trembles with light. A root changes the soil around it. A fungus reaches through a pore too small for a root hair. A wasp follows a trace of damaged green tissue. A closed stoma saves water for a dry afternoon. The forest does not need voices to be full of messages. It has leaves, roots, fungi, water, scent, shade, and time.